Welcome to Lesson 13.1, RNA. In this lesson, we'll contrast RNA and DNA and explain the process of transcription. Let's compare and contrast RNA and DNA. Make a diagram in your notes that compares and contrasts these two molecules. If you recall, DNA is made up of nucleotides, which consists of 5 carbon sugars, a phosphate group, and a nitrogenous base. Well, RNA are also made up of nucleotides, but theirs are different than DNA. The sugar in RNA is ribose instead of deoxyribose, so that's the R in RNA. RNA is generally single-stranded, not double-stranded, like DNA. And RNA contains uracil in place of thiamine. So you'll never see T's in RNA, you'll see U's. So what would U be complementary to? Well, just like A bases with T or pairs with T, U would pair or be complementary to A. So adenine is complementary or combines with uracil in RNA. You will not see any thiamine in RNA. RNA plays an important role in the production of proteins. The roles played by DNA and RNA molecules in directing protein production are like two types of plans builders use, a master plan and a blueprint. Let's use this analogy to compare the purposes of DNA and RNA. The master plan that the builders would be using contains all of the instructions to build an entire building. This is so important that the planners probably wouldn't want to let this leave their office. So this would represent the DNA inside the nucleus. The master plan is DNA, and it's safe inside the nucleus. Well, these plans need to get broken down into components to build specific parts of the building. So these blueprints are types of RNA molecules. They are replicated from the master plan, and then these messages or instructions are delivered to the people who will actually do the work. So keep this analogy in mind as we look at the types of RNA and how the information in DNA is moved by RNA and used to create proteins. Let's take a look at the three main types of RNA. The first type of RNA is messenger RNA. Sometimes this is abbreviated to mRNA. Messenger RNA is a type of RNA molecule that takes a copy of a specific part of DNA and then messages it or moves it outside of the nucleus to ribosomes where proteins are produced. Now, as you can see, this is a single strand, whereas DNA was a double strand. This molecule just has a single strand of nucleotides. So these colors here represent the nitrogenous bases of RNA. And if you recall, those bases are A, G, C, and in DNA we had T. We don't have T in RNA, we have U. The next type of RNA is ribosomal RNA, or rRNA. As the name implies, ribosomal RNA helps make up ribosomes, the site of protein synthesis. tRNA, or transfer RNA, is used to help move specific amino acids to the ribosomes where they're added to a chain of amino acids to make proteins. So let's take a look at these types of RNA in more detail. Messenger RNA, or an mRNA molecule, is a copy of the portion of DNA that will be used to make a protein. Messenger RNA carries instructions for polypeptide synthesis from the nucleus to ribosomes in the cytoplasm. Now let's recall, what is a polypeptide? That is a fancy name for a protein. Polypeptides form proteins. So we're going to be making proteins. mRNA provides the instructions on how to do that. Okay, protein synthesis occurs in ribosomes, which are made up of two subunits. The ribosomal RNA forms an important part of both subunits of the ribosome. During protein synthesis, transfer RNA molecules carry amino acids from the cytoplasm to the mRNA at the ribosome. Take a moment to create a table in your notes that identifies the 3 types of RNA and their main function. Let's take a look at how RNA is synthesized or made. The process is called transcription. The prefix trans means to move, and script or scribe means to copy. So you're going to move a copy. In transcription, segments of DNA serve as templates to produce complementary mRNA molecules. An enzyme, RNA polymerase, is used to help facilitate this. The enzyme unzips the DNA molecule. As you recall, DNA is 2 strands. The template strand is then used to create the mRNA. This occurs by base pairing. As you can see here, guanine is complementary to cytosine, so cytosine is being added to the RNA strand. Here we have thymine, which is complementary to adenine. Here we have adenine, and if you remember in DNA, it would be complementary to thymine, but we're building RNA, so we have uracil. So as you can see here, these nucleotides are being added to this mRNA strand. This strand contains the information to produce a protein. This process is called transcription. So how does the enzyme RNA polymerase know where to start and stop making a strand of RNA? Well, RNA polymerase doesn't bind to DNA just anywhere. The enzyme binds only to promoters. Promoters, which have specific base sequences, tell the enzyme where to start transcribing DNA. So let's take a look at the process of transcription that's going on here. The enzyme has unzipped the DNA strand. The template side of the DNA strand is being transcribed by base pairing. Notice that the guanine on the DNA side is pairing up with the cytosine on the RNA side. Thymine is complementary to adenine, and in RNA, adenine is complementary to uracil. When the mRNA strand is transcribed, the cell will sometimes edit that RNA molecule. Part of the mRNA molecule are snipped out. These are called introns. The remaining parts of the mRNA, or exons, are connected together, and then they get a cap and a tail. This final copy of the mRNA is then delivered to the cytoplasm, to the proteins, where protein synthesis occurs. Take a moment to summarize the process of transcription in your notes. Make sure to include an example of base pairing where mRNA is made from DNA.